Well, hello to all of you, and thank you for joining us. My name is Carolyn Thompson, and I'm the Director of Philanthropy for the University Hospital Foundation. We have people joining us from all across Canada. Well, not just all across Canada, but Qatar, Shanghai, and the Philippines. So welcome. And it is, of course, my pleasure to welcome you all to an education on COVID vaccines and their safety, the latest event in our increasingly popular virtual expedition series, where you have the opportunity to hear from and engage with some of the best and brightest medical minds in Canada. Thank you so much for joining us. Now, one quick point to tell you about this session. It is being recorded, and we will send a link to everybody that registered for this event over the next few days. We call these sessions expeditions because they take you on a journey through the many amazing areas of care here at the University of Alberta Hospital site. And the best part is they are guided by some of the truly incredible people who work here. Today, we're covering a topic that is one of the most talked about subjects in the entire world, and that is vaccination safety. To get us started, it is my pleasure to introduce to you the President and CEO of the University Hospital Foundation, Dr. Jody Abbott, a woman of vision who is as passionate about igniting change in healthcare as Dr. Terrell and Dr. Lewis are on their focus of COVID-19 research. Jody?
Thank you very much, Carolyn, and good afternoon, everyone. I think Carolyn said something that deserves repeating. Our topic today is or has been on the minds of the majority of the world's population, and that is COVID-19, and more specifically, the safety of vaccinations. If you recall last year around this time, the concern was stopping the spread or flattening the curve of COVID-19. We were introduced to the concepts of washing our hands for 20 seconds and masking, Zoom meetings, elbow bumps instead of hugs and handshakes, and of course, social distancing. Today, very little of that has actually changed, and we need to continue to be diligent. However, we may see the light at the end of the tunnel as we now have a vaccine. In fact, we have several of them. And with them has come some hope. We believe in the power of these vaccines to end the pandemic and save lives. But many people still have questions, which is why we thought vaccine safety would be an excellent topic for our expedition today. Another reason for choosing this topic are the two doctors you're going to hear about today. They bring a level of expertise, experience, and depth of knowledge that few people in the medical world can equal. We're proud to have supported Dr. Terrell and Dr. Lewis over the years in their scientific and clinical endeavors. They are true leaders in their respective areas of specialization. One of them has been credited for helping to save the lives of millions of people around the world for his work on finding a cure for hepatitis B. And the other is currently conducting clinical trials on a made in Alberta COVID-19 vaccine. They are in every way the embodiment of the boldness and tireless quest for solutions to seemingly insurmountable challenges. This forms the core of why we do what we do at the University Hospital Foundation. I'm incredibly proud and absolutely thrilled that they are able to join us today. Now, before we begin, for those of you who don't know who we are and maybe are new to us, I'd like to tell you a little bit about us. The University Hospital Foundation was created in 1962. We raise funds to advance patient care and innovation at the University of Alberta Hospital, the Mazankowski Alberta Heart Institute, and the K. Edmonton Clinic. In the ensuing decades, we have evolved into one of the most successful healthcare fundraising organizations in North America. And thanks to the generosity of our donors, we have established world-class cardiac and brain care at the University of Alberta Hospital and provide millions of dollars every year for researchers right here in Alberta to give them the resources they need to make discoveries that will change the lives of people here and around the world. Our donors answered the call yet again by joining the fight against COVID-19 and giving to the University Hospital Foundation's Emergency Response Fund, which began last spring. Our donors are helping ensure that our hospitals, healthcare teams, and all Albertans have the resources and access to research they need during this pandemic. So let's learn a little bit about our audience and how do you feel about COVID vaccines? We invite you to participate in a live survey. The results of the survey are anonymous and may show us if there are any trends within the group. We're going to allow you a few moments to fill in the survey.
Right, the results are coming in. We'll give it a few more seconds here. All right, the survey is now closed.
Okay, this is really interesting. So it looks like many of you have had your vaccine. Some are planning to, and I see some still undecided or haven't yet got the vaccine or have decided they won't. What I hope today is at the end of the presentation, you'll be able to ask any questions you might have, and that will help you to make some decisions as you go forward. Now I'd like to introduce you to our first guest, Dr. D. Lauren Terrell. Dr. Terrell served as the Dean of the Faculty of Medicine and Dentistry at the University of Alberta from 1994 to 2004. Beginning in 1986, his research was focused on viral hepatitis. His work on the development of antiviral therapy for chronic hepatitis B resulted in the licensing of the first oral antiviral agent to treat the condition. He has also co-funded a biotech company, KMT Hepatech Inc., based on the first non-primate animal model for the hepatitis C virus. He is a distinguished university professor at the University of Alberta in the Department of Medical Microbiology and Immunology, and a founding director of the Li Ka-shing Research Institute on the U of A campus. A recipient of the Alberta Order of Excellence, an officer of the Order of Canada, a member of the Canadian Medical Hall of Fame, and winner of the prestigious Killam Prize given each year to Canadian scholars who have distinguished themselves through sustained excellence and impact in their field. Since the beginning of the pandemic, Dr. Terrell has worked on antivirals and vaccines for COVID-19. He is a member of the COVID-19 Vaccine Task Force that advises the federal government on the selection of COVID-19 vaccines based on efficacy, safety, and timely availability for Canada. Folks, this is a rare opportunity to meet a living legend in any field, much less in the area that has consumed our lives for the past 15 months. I'm very pleased to welcome Dr. Terrell. Lauren, over to you.
Well, thank you very much, Jody. It's wonderful to be here. Wonderful to address the whole University Hospital Foundation audience that's here today. I'm going to concentrate on some of the work that we've done on the vaccine task force because I think it's very relevant to what we're asking about today. Can I have the next slide, please? The objectives are to understand the methods of how these COVID vaccines are produced. And there are a number of options, in which some are favored. To describe the safety of the new vaccines, they came on so quickly, people are concerned about the safety. To discuss the vaccine supply issue, we know that has been a problem for Canada. To discuss and determine if rapid views are taking any shortcuts. and to outline concerns over the emerging virus mutants and the vaccines, will they still be effective? Next slide, please. Let me first say that there has never been vaccines developed this quickly. Traditionally, they take 8 to 15 years to get a vaccine on the market. And this virus was discovered in December of 2019. The sequence was in virus identified on January 9th, and the sequence on January 24th. And some of the vaccine companies, within a week, had taken that sequence and started to make vaccines, particularly the RNA vaccines. And these were approved December 2020 and December 23rd, 2020, all within a year. Absolutely remarkable achievement that nobody thought was possible in the beginning. Sorry. Next slide, please. So I was selected as a member of 12 people who sat on the vaccine task force appointed by the federal government. And our chore was to find the vaccines for Canada that are the safest possible, the most effective, and timely access to them. And key is that vaccines are critical to controlling this pandemic. And we knew that was so important at the very beginning. Next slide, please. So what is the role of the Canadian government in all of this? The Canadian government completes the procurement agreement with the manufacturers of the vaccine. And we looked at a large number of vaccine candidates, and we selected 7. We didn't know how these vaccines would work. We didn't know about their safety, but based on the cumulative knowledge of the panel, which has been wonderful to serve on, we came up with recommendations that turned out to be pretty accurate. But Canadian government then goes out and signs procurement agreements with the manufacturers so we can get vaccines for Canada. Vaccines must pass the health protection standards of Health Canada before they can be licensed for use in Canada. The Public Health Agency, once they're licensed, is asked to distribute the vaccines out to the provinces and to the territories. And the provinces and the territories will execute vaccinations and roll out strategies for their province. After all, healthcare is a provincial responsibility. So once these vaccines are given out to the provinces, they decide how they'll be used. Next slide, please. Let me say, one of the issues we ran into is countries do have emergency use authorization. The United States, the European, and the UK all have EUAs, which means you can make an emergency authorization quite quickly. In September the 16th in 2020, a couple of months after we got started, Canada introduced an interim order, which gives the vaccines 1 year to support their access to COVID drugs and vaccines. It gives them approval for a year, but then they have to show that they are really effective after that year. So we, instead of an EUA, have used the interim order. Next slide, please. I think it's important for you to understand what an interim order in Canada means. It means you can give in rolling submissions of the data. Normally, a company has to accumulate all the data and present it at once, and then takes a few months for Health Canada to make a decision. When you have a rolling submission, it means the data can go in on a continual basis, and it means we're going to get approval quicker. And given the gravity of the situation with COVID-19, rolling submissions was very important. It also allows us to access the data that was used in foreign approvals, particularly United States, UK, and EU have been accessed their information about the approvals. So in fact, I think we get more eyes looking at the vaccines to make sure they're safe for the public. And it's an expedited review. We do not take any shortcuts on the review process. It often improves the thoroughness of the reviews as the information is shared much more efficiently between countries. And I think that's extremely important to make sure that we have safe vaccines. Next slide, please. So vaccines, are critically important. But I just want to make sure you understand that vaccines are for large populations of healthy people. It's very different to the antivirals, which are used in people with the disease. For the antivirals, people are more tolerant of the balance between safety and benefits. But in vaccines, you want to make sure they're extremely safe because they're going into healthy people. Next slide, please. So the SARS virus has on its surface those spike proteins we've all seen in different pictures. And the spike protein binds to an angiotensin converting enzyme 2, which is on the surface of host cells. And the interaction between the spike protein and the receptor on cells is the critical way that the virus eventually gets into the cell. And if you can block this interaction, this is what vaccines can do to prevent the infection. Next slide, please. This is a big virus. Most RNA viruses are 10,000 kilobases or less. This one is 30 kilobases long. Very large for an RNA virus. However, a big portion of the structural proteins is the spike protein that interacts with the cell surface. And this is the red part. And the vaccines are designed to make spike proteins. And if you get an antibody to the spike protein, it'll block the interaction with the host cell. Next slide, please. So vaccine production came into play very quickly. And the way we've made vaccines include inactivated vaccines, where you kill them of the virus and use it as a vaccine, or live attenuated, so it's a weak vaccine. These both take some time to develop. And then the new one was a vector delivery with the adenovirus, and I'll discuss that one, as we have two of the vaccines, both Astrazeneca and the J&J vaccines are of this type. And then messenger RNA vaccines, Pfizer, BioNTech, and Moderna follow this. Self-amplifying RNA vaccines, protein subunits, and we have some of these coming. We have a viral-like particles coming, and we have DNA vaccines, which John Lewis will be talking about his DNA vaccine to you. Next slide, please. So the messenger RNA vaccines have revolutionized vaccinology. The messenger, normally information flows from DNA through messenger RNA to proteins. And we've made vaccines with proteins. But if you could make it with the messenger RNA, it would be easier than making it with the proteins. So the benefits of the RNA vaccines over conventional approaches is the safety. These messenger RNAs are not infectious. They do not integrate into the host cell genome. And once the RNA has been translated, they have a relatively short half-life, and the protein is made and they disappear. Vaccine's gone. Production, vaccines can be produced very much more rapidly with the mRNA technology, and you can modify it rapidly also to adapt to the new mutants if you need to. The vaccines generate a reliable immune response, as we've clearly seen, and they're well tolerated by healthy individuals with very few side effects. Next slide, please. The delivery is a bit challenging. Free RNA in the body quickly breaks down. So they're actually delivered in a lipid particle called almost a liposome. And that's very important in making RNA vaccines very successful. And like conventional vaccines, these vaccines need to be kept in very cold storage where the RNA can break down. And storage outside the cold chain leads to some instability, and that makes it hard to deliver in some countries where cold chains are hard to maintain. Next slide, please. So the mRNA vaccines for Pfizer Bioindex, there was 2 injections, 3 weeks apart, as recommended by the developer. And as you know, we have spread that out, and I'll talk a bit about that later, to up to 16 weeks in Canada. Phase 3 studies have shown with this regime to be 95% protective. Let me say that flu is often anywhere from flu vaccines from 30% to 70%. So we were hoping to get at least 50% effectiveness in these COVID vaccines, and to see the results of 95% effectiveness was absolutely remarkable and delightful to see. Even after one dose, there's about an 80% protection for a period of time. As I said, the vaccines have to be stored at -70, but they have provided some of that technology as well. We recommended, and the government of Canada procured 40 million doses of Pfizer Bioindex before we knew it was going to be this effective. Next slide, please. This just shows you the results after the first dose. And you can see the placebo in blue, people kept accumulating and getting infection. However, after about 14 days after the dose, first dose, you saw that the infections were stopped and slowed right up. Let me point out that when you get one dose, you're not protected for at least 2 weeks after the dose. And with one dose, you're only partially protected. Next slide, please. These were delightful results to see. The second company that made mRNA vaccines was Moderna, 2 doses, 4 weeks apart. Phase 3 studies again showed 95% effectiveness in preventing it. Protects from severe disease very well. And it needs to be stored at -20, not -70. So the cold storage is a bit easier. And again, we procured 40 million doses from Moderna. Next slide, please. I just want to say that Moderna's co-founders both spent a good time in Canada. Nobar was an immigrant, came from Lebanon to Montreal, did his junior high and high school in Montreal. McGill graduate, went to MIT, and is the co-founder of this. The other one is Derek Rossi, did his education in Toronto, PhD in Toronto, also went to postdoc in MIT. So there's a strong Canadian content to Moderna. Next slide, please. And they have to be in, both of these have to be delivered in these liposomes or lipid particles. And Peter Cullis at UBC formed a company, Accuritas, and Accuritas is providing that lipid envelope to both of these vaccines. And Teresa Allen, who used to be at the University of Alberta and retired and is a professor emeritus, but has worked out in UBC since her retirement, again with Peter Cullis. Both have been very much involved in the liposomes. Next slide, please. Just showing you the Moderna after you get the 2 doses. The blue line is the protection, and the dark line is the placebo. Again, 95% protection. Next slide, please. The second group of vaccines were delivered by adenoviruses. And Astrazeneca Oxford is a non-replicating form of the adenovirus. Adenoviruses are common viruses in our environment, and we often have antibodies to them, but we have antibodies to the different viruses. AstraZeneca Oxford used a chimpanzee adenovirus vector, so we wouldn't have antibodies to it, and it would get into the cells, and it would produce the spike protein, 2 injections a month apart. And phase 3 studies showed about a 70% effectiveness in the beginning. Some of the later studies are showing higher levels than that. And it was 85% effective at preventing severe disease. The advantage of this, it can be stored in a regular refrigerator for up to 6 months. So you can get it out into remote areas a lot easier than you can with the mRNA vaccines. Next slide, please. Vaccines have to go through a number of phases before they're approved. A phase 1 is an acute safety studies in small numbers, often under 50 healthy people. Phase 2, about 600 people, where you see the immune response and see how well it produces protection against the virus. And then you go into a phase 3, which is often 30 to 40,000 patients. The advantage of this, it shows you how effective the vaccine is, and it also gives you an idea if there's any rare toxicities that might come up. These are done as double-blind studies. Neither the patient nor the person conducting the study knows if the patient is getting a placebo or the real vaccine. I want to emphasize there is also phase 4 studies, and this is after approval. We have obligations to monitor carefully all the people that get the vaccine for side effects. And I just want to say the phase 4 studies were very effective in picking up the rare clotting issues that has been observed with the Astrazeneca and the Johnson & Johnson, another adenovirus vector. In Canada, we've had 28 people develop these rare blood clots, and there have been 3 deaths. So as you know, some provinces have recommended stopping the Astrazeneca vaccine, even at the present time. I just want to emphasize the risk of a vascular event, and there's lots of those from COVID-19, is about 350 times greater than a vascular event from the adenovirus vectored vaccines. So there's still a great benefit over risk for people that have high risk of developing this disease. Next slide, please. I want to say that John Bell, who was raised here in Edmonton, very close to the University Hospital, and his father was a hematologist, worked in hematology at the University Hospital, is now the Regius Professor of Medicine at Oxford University, and he's been very instrumental working with the Jenner Institute in Oxford in developing this vaccine. Next slide, please. I also want to point out that Dr. Frank Graham from McMaster University worked for many years coming up with the cell lines and the non-replicating adenoviruses. And his technologies have been used by these companies developing the viral vectored vaccines. Next slide, please. So the stages of vaccine development, you go through the basic research, you do testing in animals, you get to good immune responses, no toxicity, you work out the adjuvants, and then you get a vaccine substance. But that then goes to a manufacturer that puts it through scale up under GMP, good manufacturing, so it's extremely safe, fill finish, and then it is a vaccine product that you can get out, and that's delivered to public health. That's a big job going from each of these stages. Next slide, please. So Canada, why was the drug supply slower in Canada? Well, we've lost most of our biomanufacturing capabilities. Large pharma left Canada. As we moved to more and more generic drugs, they decided to leave starting in 1968. Also, the United States invested $18 billion in 7 major pharmaceutical companies to get 30 million doses of the vaccine. And this has led to vaccine nationalism. The countries that have invested heavily or where the vaccines are being produced are keeping the vaccines for their own population first. And this is why there has been some slower delivery in Canada. Next slide, please. I just want to say that Canada, through our procurement, procured the most drugs of any company in the world, even more than the United States. But there is a big difference. Next slide, please. There's a big difference between procurement and actual delivery. Okay, let's just touch briefly on the concerns, variants of concern. Next slide. Viruses will mutate, although coronaviruses have a lower rate of mutation simply because they do have some proofreading ability. But viruses are always changing, and mutations are always introduced when the viruses are replicating. The sooner we get herd immunity, the sooner we get the vaccines into arms, the more rapidly we'll stop the mutations. Next slide, please. Viruses mutate, so they can, and the ones that will win out are those that become more transmissible. Viruses infections can become more deadly with mutations, and they may develop an ability to evade the immune system or immunity. These are the concerns with the mutations. Next slide, please. There are 4 major mutations we've heard about: the UK variant, the South African variant, the Brazilian, and the Indian. There's actually another couple. Next slide, please. I just want to say that it's a race between getting people vaccinated and these mutations from causing more and more infection. The UK, the South African, and the Indian mutations are certainly more transmissible than the original. Their R values have gone up, and it's shifting to younger people as older people have been fully vaccinated. We need to get people vaccinated to stop these down, shut them down. Next slide, please. Just show you in pink is the variants in Ontario predicted. In blue is the original strain, and you see how in Ontario, in Alberta, we've seen the variants become more transmissible. Next slide, please. This is Alberta. In blue is the UK variants. In green is the Brazilian variants. And this is up to, as of the last couple of days, May the 16th. Most of our variants are coming from the UK variant, and it's starting to go down. This is May 10th when it peaked and going down now. Next slide. Timing between the first and second doses. We got more people protected, partially protected by using a single dose, and we can protect more people by delaying the second dose. But by giving a single dose, we give protection. It's not ideal, but it's taken in times of vaccine shortages. Next slide, please. As of last Friday, we've seen that people who delayed their vaccine, people have been concerned, would they still get a good second response? In fact, the response, when it was delayed to 12 weeks in the UK, or in some studies have shown that the immune response is even stronger. And in many vaccines, we do have a longer interval than 4 weeks. So that may improve the duration of immunity when you get your second dose later. Next slide, please. So if we mix and match, and this is a question that keeps coming up, if you got the Astrazeneca and now we're stopped using it, what are you going to get? Well, there is a study that's going on in Oxford at the present time, and people who get the second dose of the Astrazeneca, so they get the first and the second the same, have had less serious local reactions. Some of the people who have got the AstraZeneca and then get one of the RNA vaccines have had more reactions of fever, headache, sore arm. And early studies suggest this is just being published May the 12th, results released, but we need to see them published. Next slide, please. Vaccine hesitancy, our concern is it is growing. We need to get it turned around. We want to get herd immunity to, as long as we delay getting herd immunity, we prolong the epidemic. We want to get as many people vaccinated as possible. Next slide, please. Conclusion, we have wonderful vaccines, particularly with the messenger RNA. I would say that people have said this is a miracle that this has happened so quickly. I would say there was probably 10 to 20 years, there's decades of research behind these RNA vaccines, and they've just been used for the pandemic, and that's a miracle. They will likely end the pandemic. Canadians have made lots of contributions. And next slide. We're weeks behind, but not months behind. And the immunity will last at least 8 months. The clotting issues are long-term safety. And vaccines remain very effective. So far, the messenger RNA vaccines will work against the mutants, and boosters will likely be required. Next slide. Thank you. Stop there. Thank you very much.
Thank you very much, Dr. Terrell. That was absolutely amazing. And I know everyone will have the opportunity to ask questions during the Q&A period. Our next guest is a researcher whose work in prostate cancer has earned him the Frank and Carla Sajonki Chair in Prostate Cancer Research funded by the Alberta Cancer Foundation. He is also overseeing 2 major clinical cancer-related studies in 2021. He is a University of Alberta professor in the Department of Oncology, Division of Experimental Oncology, and the founder, CEO, or Chief Scientific Officer of 6 spin-off companies that are developing treatments for cancer, infectious diseases, and age-related diseases. He is at the forefront of pandemic research. His biotechnology company, Entos Pharmaceuticals, is currently doing human trials on a DNA-based vaccine for COVID-19. Working with an international multidisciplinary team, including Entos Pharmaceuticals, the Lewis Lab has been awarded $4.2 million from the Canadian Institutes of Health Research to bring a COVID-19 vaccine to phase 1 clinical trials. So as you can see, we have another great Canadian making strides in this area. Dr. Lewis, the floor is yours.
Thank you so much, Jody. That was a wonderful introduction. And I just want to say, it's fantastic that the University Hospital Foundation is putting an event like this on. It's a topic that's on the top of everybody's mind. And thank you so much, Lauren. You did it's just a really fantastic job. Your leadership in this area in Canada, is just remarkable. Really appreciate all the work you've done. So I wanted to give you a slightly different perspective, talking about our vaccine efforts from the perspective of a cancer researcher now working on a COVID-19 vaccine, and really approaching the safety aspect from the very beginning, and how do we develop a vaccine in Canada, and what are we thinking about when it comes to safety, and given the fact that we have this highly compressed timeline or this urgency to move things forward, how are we preserving the utmost safety of these vaccines? And so next slide, please. So I guess go back about a year and a half ago, as Jody mentioned, I run what we call a translational prostate cancer research program at the University of Alberta. And in this program, we're doing fundamental research on cancer, but thinking about what is most deadly about cancer, how can we develop better tests for cancer, and all the time we're thinking about line of sight to the patient. How can things we discover in the lab be applied in the clinic for the benefit of patients, whether they be improved tests or therapies. And so we believe very strongly in doing clinical trials. We've, for instance, created the Alberta Prostate Cancer Research Initiative and created a large biobank and biorepository of specimens that we're using to test new therapies and new tests. And we've really been really active spinning out companies, as Jody mentioned, to try to bring the most promising technologies into the clinic for the benefit of patients. Now, one of those efforts is in the area of personalized medicine for cancer patients. We're very active in developing gene therapies and personalized cancer vaccines for cancer patients. And so we were, right at the beginning of the pandemic last year, we were right in the perfect position with the technology that could be brought to bear on developing a cancer vaccine that was pretty, very complementary to the vaccines that Dr. Terrell mentioned. Next slide. So in 2016, we launched a company called Entos Pharmaceuticals, and very much like BioNTech or Moderna, we've developed a technology that initially was designed for cancer gene therapies and personalized cancer vaccines to be able to basically create these genetic medicines that can treat, very specifically diseases like cancer, but can also be extended to other diseases such as age-related diseases and childhood genetic diseases, and as we learned, also vaccines. So I guess one of the key differentiators of Entos's technology is that it can deliver RNA, but can also deliver DNA, and I'll get into a little bit into that as we went through our thought process. So Entos has a large research and development facility in Edmonton. We are working very closely, with a number of other Edmonton companies, including the University of Alberta's Advanced Cell Therapy Manufacturing Center, which creates sort of clinical grade medicines. And so we just have a great team, a lot of support locally. Next slide. And so I mentioned the Alberta Cell Therapy Manufacturing Center. So with our relationship with them, and also a relationship with a Vancouver company called Precision Nanosystems. Actually, you mentioned Peter Cullis, so founded by Peter Cullis. This great Canadian technology development has allowed us to take discoveries that we make right here in the lab in Edmonton to be able to scale them up, manufacture them in the very safest way to make a clinical grade vaccine or gene therapy, and do early stage clinical trials and eventually produce vaccines for Canadians. So with with this partnership with Precision Nanosystems and the ACTM, we actually right now have built out manufacturing capacity to produce about 400,000 vaccine doses per month right here in Edmonton, Alberta. And we're hoping to use this as a basis to build a full commercial manufacturing facility in Alberta as we move our vaccine and we see other Canadian-made vaccines move toward commercial manufacture. Next slide. So we haven't done this in a bubble. Dr. Terrell mentioned that it's really been an unprecedented international effort to both learn about the vaccine, but to bring to bear all of our efforts and our technologies and expertise to be able to generate what we call countermeasures, vaccines, therapies against COVID-19. We have a truly pan-Canadian vaccine all the way from Vancouver. We're working with Vito Intervac. We have a great collaborator there, Alison Kelvin. We're working with actually the Ottawa Biotherapeutics Manufacturing Facility in Ottawa, filled our vaccine into vials. We're working with the Canadian Centre for Vaccinology in Halifax. So the PI, principal investigator of our trials, called Scott Halperin, and of course, Entos co-founder Roy Duncan, who's a microbiologist, discoverer of the technology we're using. And we have this fantastic team at the University of Alberta and at Entos Pharmaceuticals that have been pretty much working night and day for the past year and a half to get this vaccine moved through the stages to clinical trials where they are today. Next slide. So I think it's not a secret to anybody that we're in a really unprecedented situation that we couldn't really predict it could have happened. We just passed a pretty grim milestone this week, 25,000 Canadian deaths. The impact, on people, mortality has been unprecedented, but also on our economic system, our daily life. It seems like it's lasting forever. And as Dr. Terrell mentioned, traditional vaccine development takes a long time. I'm actually being optimistic here. It can take, the averages I think have been some 12 years to bring a vaccine all the way from discovery to approval. And there are a lot of diseases where we tried to make vaccines and failed. So HIV is a great example, but SARS-1 is another example. We weren't able to create an effective vaccine against SARS. But I think something significant has changed in the last 5 to 10 years. And this is the development of really innovative new platform technologies that we've seen have allowed us to create and develop and approve effective vaccines in a record time. And I'll explain to you over the course of this talk that the record time is not due to the fact that we've cut any corners. It's the fact that we have ready to use plug and play platform technologies that we were applying to COVID-19, but now can be applied to many other problems that plague us as well. And I'll just mention as well, vaccine development can be a tough slog, can be very expensive to do. Commercially, it's tough potentially to make money in vaccines. But vaccines are really, really critical to our control of disease. They're most efficient and most cost-effective way to prevent and eradicate disease. Next slide. So I just want to talk quickly about how SARS-CoV-2 infects, because it's really important in the way we design vaccines. So SARS-CoV-2, as Dr. Terrell mentioned, it's an RNA virus. It interacts with a receptor called ACE2 on a variety of different cells in our body, mainly in the respiratory system. So we breathe it in through our nose, through our mouth, can get down to our lungs, and basically it gets inside the cell, and its RNA inside the virus then creates the proteins that allow it to multiply and then be released again in great numbers from that same cell. So it continuously cycles in and out of cells. This is really important when you're thinking about making a vaccine, because this is typically how your immune system will see it and begin to mount an attack. Next slide. So I'm just going to quickly go through the technologies, because traditionally vaccines have been developed really using the virus itself or a piece of the virus. So the idea of making a vaccine is really to create a wanted poster to show your immune system who to avoid. So if you see this candidate, attack them right away. They're a dangerous player. And so a vaccine basically provides those wanted posters to the immune system. And the way we show the immune system, either the whole virus or a piece of the dangerous virus, we can do in a number of different ways. So traditionally, we've actually taken the virus itself and either what we call attenuated it, so made it weaker so it can't kill you, or completely inactivated it. So the structure is still there, but it can't infect. Or we take a piece of that virus, one of the proteins in that virus, and show it to the immune system. The key here is that we're only showing a very, very tiny amount. So you get a shot in your shoulder. It's just a really, really tiny amount of either the proteins, and as I'll tell you, maybe the genetic material of the virus, that then creates this wanted poster that your immune system can recognize, so that when you're actually exposed to the real virus, your immune system's trained, it's ready to go, it can kill it immediately. And so there's been a rise, as I said, of these genetic medicine technologies over the past 5 to 10 years that allow us now not only to generate vaccines that have the whole virus or protein subunit, but actually just deliver the instructions to your body, so that your body, your own cells, can read those instructions, make a piece of the virus, so that your immune system can immediately respond to it, create those antibodies and T cells that can protect you against a potentially deadly infection in the future. And there's a number of different ways you can deliver those instructions. So in the body, our DNA sort of contains the blueprints for all the genes. And so DNA delivered as a vaccine will make RNA, which will make protein, which will alert the immune system. Or with RNA vaccines, we can sort of just deliver the RNA intermediate, which can provide a strong protein response. But again, RNA is less stable than DNA. So there are pros and cons to each. On the other hand, we can take, as we see with the adenovirus vaccines, like the Astrazeneca and Johnson & Johnson vaccine, we can take the instructions and stuff them inside an innocuous virus, like a cold virus, adenovirus in this case, to make those, to show that wanted poster. Next slide. So I think the key, though, to producing an immune response is really to really ignite and stimulate two sides of the immune system. So the first is to elicit antibodies against the pathogen or coronavirus. So this creates different antibodies that recognize the virus, that block its ability to attach to this ACE2 receptor and infect. On the other hand, too, we also have a cell-based response. Just hit forward. Just go all the way to the end, please. Perfect, one more, awesome. So we also, so the other side of our immune system, the adaptive immune system, is the creation of T cells or killer T cells. And so, so while antibodies block the virus from infecting cells, T cells then go around and surveil and look for any cells that are becoming factories to make more virus, and they kill those cells very specifically. So producing an antibody response and a T cell response, I think both is critical, because antibodies, neutralize the virus, when you have the early infection, so it protects against viral entry. But these antibodies may be short-lived, so 3 to 6 months. And the antibodies can be very specific. So if the virus, mutates and has variants, it is vulnerable to escape from these variants. On the other hand, T cells much more broadly recognize the virus. They're very, very long-lived. So we can find circulating T cells that recognize SARS-1 in patients that recovered from the first SARS epidemic. And T cells also can target the very, very most conserved parts of the coronavirus to provide potentially protection against all the variants with a single response. Next slide. So, Lauren really, set the frame for this. We had an opportunity at the beginning of this pandemic. We got, unprecedented ways to sequence the virus, to learn about it. Scientists from around the world really started to share information. So information about the virus widely circulated. And so we had this opportunity to combine these really novel, innovative genetic technology platforms with this information that was coming out quickly. And we saw, unprecedented speed in the development of new vaccines. Again, though, we've failed in the past to make some vaccines. We've actually made some challenges with vaccines. So, for me at the beginning, when we got this CIHR grant, we really focused on going fast, but making sure we avoided pitfalls of previous vaccines. Next slide. So one challenge that we learned in the 1960s was seeing that certain vaccines can create actually enhanced disease when you're vaccinated. And so we saw with the respiratory syncytial virus vaccine that when you just took the whole virus and you inactivate it with formalin and vaccinated individuals, often what you'd find when they actually acquired the infection was that they had more severe disease than unvaccinated individuals. And it turns out when you formalin fix or inactivate the virus, you actually change the way it is displayed to the immune system in this wanted poster. And actually that created an unwanted immune response that created elevated cytokines. So they had a very potentially dangerous situation where the infection created the situation where the immune response was being stimulated more than it should be and was creating a dangerous situation. So we wanted to avoid vaccine-enhanced disease. Next slide. The other thing that we can encounter is that your body, is recognizing this virus, it's creating antibodies against it. And what we know is that only the antibodies that can block the coronavirus spike protein from interacting with ACE2 can block infection. But your body also produces, it's trying different things. It's producing antibodies against all different parts of the coronavirus that it can see. And some of these antibodies can actually stick to the coronavirus and pull it into cells. And so if you get too many non-neutralizing antibodies and less neutralizing antibodies, this can actually help the disease along. So we wanted to think about that in the design of a vaccine. There's also this other similar situation that we've seen with certain vaccines where antibodies can actually cross-link different pieces of the immune system to activate, again, this increase in cytokines and potentially create inflammation and exacerbate the situation more than it needs to be. So again, we want to very, very selectively and specifically show the immune system the piece of the virus or in a way that it produces an appropriate response when you're actually infected with SARS-CoV-2. Next slide. So I guess the take-home message for all this is that because of the way SARS-CoV-2 and other viruses get into a cell, replicate and then are released again, genetic vaccines are really the ideal way to produce an immune response. And we've really seen this through the success of the RNA vaccines, because with an RNA or DNA vaccine, they're taken up into the cell, they provide the instructions for the cell to make the spike protein or make other proteins from the coronavirus. These are either released out of the cell, where then they can be recognized to produce antibody responses, or they're chewed up by the cell and displayed on the surface so that T cells can come along and say, hey, this is a foreign invader. Let's train ourselves to be able to recognize and kill any cells that are infected and are becoming factories for the virus. So this, genetic vaccines provide this really nice two-pronged response. The other thing we found with genetic vaccines is that we can also incorporate, little nudges to the immune system to push the immune response in a way that's going to maximize the protection and minimize the chance of any unwanted responses. Next slide. And so, if there's one thing we've learned from this pandemic, that we're in an age of genetic medicine. So we've sequenced the human genome, we can sequence the genomes of viral invaders very quickly. So we can very rapidly design genetic medicines that can counteract them, including, these RNA vaccines. And as I'll say, viral vaccines. So if we're thinking about delivery instructions to cells in patients, there are really two main ways to do it. And Dr. Terrell, introduced this perfectly. So there are viral vectors. So these are the adenovirus. This is an innocuous cold virus that we put a piece of the coronavirus instructions inside. And so these are being used for a lot of different applications, including approved gene therapies and now vaccines. And so, but they have challenges. As Dr. Terrell mentioned, they're a virus themselves. So they elicit or stimulate their own immune response, which can make it challenging to give multiple doses. On the other hand, you have these, lipid nanoparticles, partially developed, pretty much all developed in Canada and, by some great Canadian scientists. And they're also an approved systemic therapies and vaccines. They do a great job of delivering RNA. But they actually, lipid nanoparticles don't do a great job of delivering DNA. So I guess the take-home message from this slide is that viral vectors deliver DNA, lipid nanoparticles deliver RNA. And at Entos, we thought, what if we could harness the power of the specific part of the virus that delivers the genetic material, but package it in a lipid nanoparticle, because they're much easier to manufacture, they're much easier to scale up, and they can be dosed multiple times. Next slide. And if we're thinking about a world where we need boosters, it's really essential that we're able to give multiple doses. So at Entos, we developed, working with this other great Canadian scientist, Roy Duncan out in Halifax. He discovered this kind of fusion protein from a virus that actually doesn't even infect humans, that's able to allow fusion of nanoparticles with cells and allow for the delivery of RNA or DNA. So Entos has been working again on this platform, working on the manufacturing of it to be able to generate cancer vaccines and gene therapies. So we've developed a platform now that actually is quite scalable. So again, with a single manufacturing unit at the Alberta Cell Therapy Manufacturing Facility, we can create hundreds of thousands of doses in a single run. And so we decided last March to apply this platform to the development of COVID-19 vaccine. Next slide. And so, we knew at the beginning these RNA vaccines were likely to work, but we knew that RNA would have challenges. We knew stability would be a challenge because they need to be kept really cold. DNA, on the other hand, can achieve the same goal, but it's stable in the fridge for over a year. It's stable at room temperature for over a month. And DNA potentially can be effective after a single dose. Again, RNA does it too, but DNA also produces this very strong T cell response, which we think is essential for protection against variants and duration. And thinking about emerging mutants and these emerging variants, we designed a second candidate, specifically for protection against variants. And again, I mentioned the scalable manufacturing. Next slide. So we developed two candidates that we're bringing forward to the clinic. VAX-001 is in the middle of a phase 1 human clinical trial at the Canadian Centre for Vaccinology in Halifax. Biologically, it's very similar to the RNA vaccines. It contains the full-length optimized spike protein, but encoded in DNA, so it's really super stable to be kept in the fridge and transported sort of normally in the cold chain like a flu vaccine. And then our second candidate, we actually used artificial intelligence to look at all of the proteins in coronavirus, including, most importantly, those proteins that are conserved between all the coronaviruses, so that we can protect against the emerging variants, plus any emerging coronaviruses that come out in the future. And so this contains 34 pieces of protein from both spike, but also the envelope and membrane proteins from SARS-CoV-2. Next slide. And so, we've done a lot of work to bring these vaccines to the clinic. So both vaccines, at every dose we looked at it in our preclinical studies, produce an antibody response. When we look at their ability to block infection, they produce a potent neutralizing antibody response. Most importantly for us, we really wanted to stimulate a strong killer T cell response that was appropriate. So we saw a balanced killer T cell response. We didn't see any evidence of any skewed response that would result in antibody-dependent enhancement. And we were able to produce T cells in immunized animals that were really effective at killing these SARS-CoV-2 infected cells. Again, VAX-001 that's now in the clinic protected animals from infection, either with a single dose or 2 doses. So we're really hopeful that this will be effective after a single dose. And this was further bolstered by data for VAX-001 that stimulated similar levels of these neutralizing blocking antibodies with a single or 2 doses, basically exactly the same response. Next slide. So currently we're in a phase 1, phase 2 clinical trial. Again, we made the clinical grade vaccine at the Alberta Cell Therapy Manufacturing Center. We filled it into vials in Ottawa. We sent it off to Halifax. It's truly pan-Canadian effort. In the phase 1, we'll be looking at 72 participants at 2 dose levels. So dose levels that are similar to the RNA vaccines in healthy young adults and then the elderly. And in as we move into phase 2, we'll be looking at, again, these 2 dose levels, but looking at single dose and 2 dose, the ability to produce this T cell and antibody-based response. And if everything goes well, and we're able to open trials in locations that still have enough individuals that are unvaccinated, we're looking forward potentially to approval before the end of the year. Next slide. So I'll just end off with a couple slides. Just thinking about from the perspective that I presented about safety. So I guess the one thing we were somewhat concerned about a year ago was this vaccine enhanced disease or antibody dependent enhancement. So we have seen absolutely no evidence of any of that for any of the approved vaccines. This doesn't surprise me at all because all the approved vaccines are these genetic vaccines. And because they make the antigens inside cells, we don't expect to see this and we haven't. And I'll just say, we've been rushing as fast as we can to move things forward, but our vaccine development in general has been dramatically accelerated, not due to cutting any corners, but really due to three things in my mind. So the rapid sequencing and sharing of the science around SARS-CoV-2, so we could get it right the first time. This is really essential. These innovative new vaccine technology platforms, whether they be RNA, adenovirus, or our Fusogenics platform. And as Dr. Terrell mentioned as well, massive upfront investment in vaccine development and manufacturing that allowed teams to really execute many of the tasks that are normally done one after the other in parallel, but without compromising safety. This is really key. And because they got it right the first time, they were able to go right from beginning to end to have highly effective vaccines. So I'm happy to say, and this has been borne out by a ton of data, the approved vaccines we have, the choice of taking are extremely effective. And so that really makes the math around deciding whether to take them easy, because it makes them all far safer than actually contracting COVID-19, even if you're in a relatively low risk group. Next slide. And so, I hope we're close to the end, but I think, we have to be prepared for what's coming. We've been racing, but I think we need to be ready to continue racing for a while. It's more like a marathon. If we're going to get control of this virus, we really need to vaccinate the world population. So we're looking at 16 billion doses or more needed worldwide. We've already seen novel variants emerge. They've escaped the protection of some of the vaccines. It looks like the RNA vaccines are pretty robust against the variants, which is really encouraging. But I think, given the fact that there's so many people infected with this virus, it's giving the opportunity for it to evolve, and we're likely to see it become endemic and require yearly boosters. Personally, I think, and this is the reason why we're still going full speed, I think it's essential that Canadians have access to a domestic supply of vaccines and boosters. And the federal government has made key investments to ensure that this is the case. And I know the Alberta government is considering doing the same thing. And I think, I believe, like many people do, that widespread vaccination is our best path back to a normal life that I'm certainly looking forward to, and looking forward to getting together with many of you. Thank you so much for your attention.
Thank you so much, Dr. Lewis and Dr. Tyrell. Like everyone here, I am looking forward to hearing more from you during our Q&A session. My name is Prince Osei. I'm a member of the development team here at the University Hospital Foundation. And I am going to be the moderator for the question and answer period of this expedition. And so my first question goes to, I'll start off with Dr. Tyrell and then jump off to Dr. Lewis right after. Would you take the vaccines that are available today? And would you encourage your family members to take it, your daughter, your granddaughter?
Yes, I would, and I have. And I take the vaccine that was available at the time that we got the vaccines. And just because vaccines are still much better than the side effects of the vaccines, if they protect you well from disease. And I have my children, and where I've been able to get them vaccinated. The son is in healthcare delivery and was vaccinated early. I was vaccinated, my wife was vaccinated. We qualified from our age and we took whatever was available. Thanks.
Thank you. Dr. Lewis.
Yeah, absolutely. Yeah. So both my wife and I are vaccinated. My wife's a physician, so had early access to the vaccine. And now our 13-year-old daughter is going to be eligible soon as well. So I think, definitely I agree with Dr. Terrell. The vaccines that are out are safe and available and key to the curbing of the spread.
Thank you so much. So we do have a lot of matters on the webinar today, and there's questions on the efficacy and safety of the COVID-19 vaccines for pregnant women. There's also, would it interfere with pregnancy? And for nursing mothers, should they be worried that it could pass through the breast milk to their babies?
It's very clear that pregnant women do not do well with COVID-19, and more of them a bigger proportion have ended up in ICU. And it's scary for pregnant women. So definitely pregnant women should be vaccinated. This does not interfere with the baby. It does not pass the placenta. And it does not, the vaccines will never get into the breast milk to cause any problem with the baby. So no, the pregnant women need to be vaccinated.
Thank you. Dr. Lewis, there's been a lot of questions about cross-vaccination, mix and match. This is where you either get AstraZeneca at first, and then you get another, perhaps Pfizer. What do you have to say about that?
Yeah, so Dr. Terrell mentioned a little bit in his talk as well. So I think, if in a world where we have limited availability of vaccines, or you find an opportunity to take a different vaccine as a booster, I think the biology of, and I'm speaking as a scientist, of course, the biology of the vaccines are all very, very similar. So I think, and we need to think about side effects as well. Side effects like fever, side effects like, feeling tired the next day. These are good side effects. This means your immune system is being stimulated. So I think we really have to, this is kind of a flu that we opt to get to avoid a much worse thing down the road. So I think, the evidence would suggest, so I mentioned the viral vaccines like adenovirus. I think the more doses you get of them, the less effective they are over time. So I think if you take an adenovirus vaccine and then you take a RNA vaccine, you're likely to see stronger side effects, but that's because you're getting a stronger immune response. I don't know if Dr. Terrell, you want to add to that?
Just add that we don't have all the data yet, and the studies are being done. They're being done at Oxford University, and the first part of that suggests that the RNA vaccine, the AstraZeneca followed by an RNA, gave you a stronger reaction in the arm and local reactions than if you got another AstraZeneca vaccine. We don't know the immune boosting, the antibodies from that study, but they should be ready within 2 weeks. We also know that there have been other studies that have shown that they are tolerable to mix the vaccines, but we need more data on that.
Thank you. And now this question goes to you, Dr. Tyrell. Canada has approved AstraZeneca, but the US has had it in waiting for many months. Do we know what they are waiting for in order to give approval? Thank you.
Not 100% sure why the US hasn't approved it, but I think that the fact that there has been some clotting side effects associated with the adenovirus vectored vaccines has delayed the approval. And I would think that, and they have had easy and better access to the mRNA vaccines, both Pfizer and Moderna. Moderna, of course, is centered in the United States, but Pfizer has had very good supply in the United States. So the necessity to approve the vaccines has been lower in those countries, in the US.
I don't know if you want to answer this one, Dr. Lewis. Someone is asking, how long after you've tested positive with COVID should you wait to be vaccinated?
Yeah, so that's good. Not my area of expertise, but I imagine that obviously, and for our clinical trials, you were excluding anybody who's had previous COVID infection. I would say you definitely want the infection cleared. I don't know, Dr. Terrell, if you have anything to add to that.
Yeah, no, we've actually done some work in my lab on this, and it's been very interesting. We were very early to see, through cooperation with AHS and the Precision Lab, that everybody who is getting vaccinated, we got some, on 100 patients, we got samples to look at. And people who had a history of COVID-19 who got vaccinated with the first dose showed a very strong response in the immune system. And in fact, you probably didn't need the second dose in those cases because your response was so strong after one dose. So there's clear that it doesn't hurt to get the vaccine after you've had COVID, probably 3 weeks after, and you'll get a strong immune response that'll give you even better protection. And I would say that convalescent sera has a variable amount of antibodies neutralizing, and people who get a vaccine usually has a higher level of immunity than those that recover from the virus. Surprisingly, but that's what the studies are showing.
So this question is from one of our participants, and it's asking, many Albertans have been given the first dose of the AstraZeneca vaccine and are waiting for a few weeks for the second dose. If Canada and other developed countries have suspended the use of the above vaccine, what is the option for the second dose for those who had the first dose?
John, you want me to say, or do you
Go
want
for it.
Go for it. I'll just say that we do have some AstraZeneca vaccines still in Canada that in the provinces are using at the present time. But I think it'll be very important to see the results coming out of the studies, both in Oxford and other studies, where the boost is with AstraZeneca or a boost with another. And if the boost with AstraZeneca is probably very safe, you haven't got a problem with the first one, you're probably getting a good result with the second one and less chances of the clotting coming up with the second dose. And so I think we'll wait and see those studies that should be out very shortly, but it's probable that you would see, it's possible you'll see the provinces open up the vaccine they have to use the second dose with the AstraZeneca, because you think this would give you a good protection.
Dr. Lewis, when do you expect vaccines to be available to children? And what are the concerns for the vaccine for children that are different from adults?
Yeah, so I mean, the order that we're looking at testing vaccines in patients is really more of a logistical thing. So we now know, Pfizer's done a large study in the age group 13 and older, and this has been approved. So I think as we expand these trials and get more data on different age groups, we'll see these age ranges increasing. Again, little humans are smaller, lower weight, maybe getting the same dose. So looking carefully for adverse reactions. I think, the data is available for younger adults is that they have actually fewer side effects than some of the older people, and then have a very, very strong immune response. So I think the data looks really good, and I'm optimistic for the child, and obviously children can still transmit the disease. So they'll be a key part of obtaining protection.
Thank you. And the next question goes
Say
back.
that some of the provinces have already approved 12 years old, including Alberta and older. And Pfizer Biotech is doing the studies in children from 6 months up. So those data should be out soon.
And could we be looking at an oral COVID vaccine?
Not that I'm aware of.
Dr. Lewis?
Yeah, so I'm aware of a few oral vaccine efforts. I know Moderna is working on an oral vaccine formulation, although I don't know how successful that is. There's a Canadian company, Simvivo in BC, that has an oral vaccine. They're testing currently now in phase 1 clinical trials. Our platform, we also have an oral formulation that we're advancing. So it's going to take a little bit longer than the standard flu shot type vaccine, but I think absolutely I think in the next year or two we'll see opportunities come up for oral coronavirus and other vaccines.
Dr. Tyrell, you mentioned the notation that you're continuing to monitor. Is it correct that this monitoring period must continue over a period of 2 years to know with certainty the consequences of the vaccines, and are these studies being peer reviewed?
Well, it's very important that any new drug or new vaccine we bring on, that we see and continue to monitor for rare side effects that occur when you go into a full population. This is always important, and they go on for at least 2 years to monitor what the effects are of these, if there's any rare side effects that might come out later. And that's standard now in the phase 4 studies, where it's compulsory to report any new side effects that you think possibly could be related. And then careful analysis of this, you start to see whether this is true or not. And that system works well. As I say, the whole thing around the clotting problems with the adenovirus vector vaccines has come out through the phase 4 studies.
So the questions keep coming, but we ran very out of time. So this question goes to both of you. If you had a crystal ball, what do you think our lives will look like 1 year from now?
Go ahead, John, or I don't mind.
I can go first, sure. Yeah, no. So I think 1 year from now, I think in North America, we're going to be living pretty much normal lives. There may be specific situations where we still need to wear a mask, and I think overall the population is going to be much more cognizant of spreading things. And I think we'll be looking at probably year or every season taking a combination flu shot, coronavirus shot, and that should keep the variants under control.
Thank you. Yeah, I believe we will be able to travel, we'll be able to get out, we'll be doing things much as we did before. I wouldn't be surprised that a number of countries would have vaccine passports. That is an indication that you've been vaccinated. I think this is going to happen. And I would think that we'll have to look at this, how we get, if people want to travel, they may need that. I think that we've learned a lot. You know, the technology of the mRNAs is going to be applied much more broadly, whether it's in cancer or whether it's in other infectious diseases. We've seen it come up with the COVID crisis, but it's going to have lots of application. Today, Canada announced a new company is coming in to work in Canada, in Toronto. Resilience is $200 million to set up a plant for making the mRNA vaccines in Toronto. Well, so I think that you're going to see more and more of that happening. I'm also very interested to see how climate change was affected by COVID. How rivers cleaned up, how climates and the cities cleared up. Maybe that's an indication that we can do more about climate change than we thought we could. Used to always hear it's impossible to move that quickly. However, we moved very quickly during the COVID crisis.
Dr. Terrell, Dr. Lewis, thank you very much for your time, for your expertise. This has been fantastic. I want to thank our audience, our participants that joined us today. That is all the time we have for our questions. I will turn now to my colleague, Caroline.
Thank you, Prince. And wow, what thought-provoking questions, answers, and presentations. On behalf of the University Hospital Foundation, including Dr. Jody Abbott, Prince, Emily, and myself, and of course, our very special guests, Dr. D. Lauren Terrell and Dr. John Lewis, thank you for your time and your active participation. We hope you found today's session informative. Within the next few days, we will send you a short survey, and you can let us know how we did and perhaps what other areas you'd like to explore with us. We will be sending you a recording from today's expedition. If you want to learn more about us, please reach out to a member of the philanthropy team or visit our website at give2uhf.ca. Continued community support is vital to continue the work of doctors like Dr. Terrell and Dr. Lewis. To date, our donors have contributed nearly $840,000 to the University Hospital Foundation's Emergency Response Fund that is providing invaluable support for COVID care at the University of Alberta Hospital now, and will support research in this area for the future. Thank you for joining us today. This concludes our formal presentation. Our next virtual expedition is on ALS research and will be held on June 24th at 4 p.m. You will have the chance to meet two doctors who are leading the way in understanding the causes of ALS and developing unique treatments for this devastating disease. You won't want to miss it. And finally, we leave you with a 1-minute video entitled Believe It. Thank you, everyone. Have a nice evening.